But thank you all for being here for the Northwest Regional Invasive Species and Climate Change Management Network Summer Webinar, Something Warm in the Water, Temperature Pathogen, and Invasive Species Impacts on Freshwater Fishes. I'm Eva Kohlberg, and I'm the coordinator of the Northwest RISCC, as well as your facilitator for today. And I'll give a more thorough introduction to our speakers, Ian Tattam and Jonny Armstrong, after a little bit of housekeeping and introductions. So first and foremost, we are recording today's webinar, and we'll be posting that to our website for Northwest RISCC and our YouTube, and you'll receive a recording link after it's posted and live as well. But for the duration of today, if you're attending live, you can interact by asking questions in the chat, and those can go to everyone, or you can direct message your hosts if you have specific tech support questions. We have Darcy Gould, who's the communications manager for Northwest CASC, who'll be helping field any tech support. But in addition to asking questions of our speakers, which will have some Q&A time after each talk, you can show your appreciation for what they're saying via the reactions button at the bottom of your screen. There also is a little caret drop-down menu by that button if you wish to hide all of the reactions of other attendees. Just if you find that distracting, you do have the power to control what you're seeing there. And just a little bit of an introduction to what is Northwest RISCC. We are a partnership of regional agencies and organizations dedicated to helping practitioners address the nexus of climate change and invasive species. And that includes a range of life forms from plants, animals, pathogens. We cover a region that spans the Oregon, Washington, Idaho central area, as well as surrounding states and provinces. And we're a program of the Northwest Climate Adaptation Science Center housed at the Climate Impacts Group at the University of Washington. I do also want to give a plug for an event that not just Northwest RISCC, but other RISCC networks from throughout the continent and the Pacific Islands regions are helping bring together in December. This will be an online event on international invasive species and climate change issues, and will be themed around the concept of better together, empowering people and partnerships. So you can find out more at that link there. I'll also drop a link in the chat. But I also want to encourage you to, if you are a manager with a success story or an early career researcher, consider submitting an abstract for our invited talk sessions. And so submissions close on the 20th of this month. So coming up, there's a link in the chat for that as well, because we do want this to be sourced by real life stories from folks of a whole wide range of expertise, from the experts at the top of their field to the people who are just coming up and who are on the ground and really have that expertise as well. So with that being said, I'd like to introduce our first speaker today, who's Jonny Armstrong. He's the PI on the project that he'll be talking about, as well as an associate professor at the Department of Fisheries, Wildlife, and Conservation Sciences at Oregon State University. And he grew up in Southern Oregon and has long been fascinated by the region's fish. He did his PhD with the University of Washington Alaska Salmon Program and was a Smith Conservation postdoctoral fellow at the University of Wyoming. His research group integrates behavior, physiology, and landscape ecology to inform climate adaptation planning, and his work in the Klamath Basin benefits from collaboration with ODFW, the Klamath Tribes, Trout Unlimited, and others, and is supported by the Northwest Climate Adaptation Science Center. So with that said, Jonny, I'll turn it over to you.
All right, let's see if I can do this. All right, does that look okay?
Yes, thank you.
All right, well, thanks so much for having me. Thanks everybody for turning out. I'm really excited to share this work with you. And today I'm going to be presenting on behalf of my co-PIs on this project, who I want to just real quickly acknowledge before I get going. And it includes, what I really love about this team is they really have complementary tool sets, and we kind of come together and have this kind of synergetic collaboration. So we work with Julie Alexander, who's in microbiology at OSU, and she's a disease expert. And if she was here, she'd be shaking her head anytime I try to pronounce the the scientific names of these pathogens that I'll be talking about. And Melanie Davis and Jacob Dickey are both spatial ecologists and excellent fish ecologists as well, who kind of did the statistics on this project. And then Jordan Ortega is a graduate student. He's still a graduate student with me, and he works at the Klamath Tribes, a former Northwest CASC fellow. And he made a lot of the observations that inspired this project, and he did the fieldwork and analysis on this project as well. And of course, I think it was already mentioned, but we really appreciate the funding opportunities from the Northwest CASC that made this project possible, and really the topics that they fund inspired us to do this work. All right, so the main course of this talk is going to be water temperature, pathogens, and the distribution of salmonids in the Upper Klamath Basin. But I want to spend maybe the first third of this talk kind of introducing the idea of the different effects that invasive species can have in ecosystems in the Northwest, and how some of the effects have been overlooked. Okay, so I'm sure if you're on this call, you're probably interested in invasive species, and pretty much if you do science or management conservation anywhere in Oregon, you're probably having to deal with some invasive species. And one of the ones that I think about a lot and that Ian's going to be talking about is smallmouth bass. We also have like barred owls, bullfrogs, a whole variety. And one of the things that can make it complex to try to manage them or understand and anticipate their impacts, they can have a multitude of effects. And one way is I think this applies to both the owls and the bass here, but they can be, well, with owls, I believe they can both be a competitor, and they can also hybridize with native owls, barred owls and spot owls. With bass, they can both gorge and chow down on native endangered juvenile salmon, but at juvenile life stages, they can also compete with them. So one type of complexity is if we're trying to think about the interactions between an invasive species like species 1 in this bubble and a native species 2, is that this arrow can represent multiple things. It can be predation, competition, and hybridization. So this is one way in which we can have these complicated effects. But there's a whole other dimension of complexity when we consider that these two species don't exist in isolation, but instead occur in the context of all the other species in the biological community. And so you can have something that we call indirect effects. And I'm going to introduce just a kind of classic kind of indirect effect to start. So imagine we have 3 species we're considering. And let's just consider predators and their prey. So say species 1 is an apex predator, and it has this top-down predation effect on species 2, which is maybe a mesopredator. And then that predator eats some species that's lower in the food chain. Well, if this top predator is having strong negative effects on this intermediate predator, it can actually have an indirect positive effect on whatever that predator is eating. And this is the basis for trophic cascades. And while this might seem like it's just some academic thing that basic science people are interested in that I might teach in my fish ecology class, this has really real-world implications. For example, the loss of piscivorous fish in lakes can trigger algae blooms that affect water quality and human health. So these potentially can have big effects that matter to humans. And I should be staying on topic here and getting to my Klamath Basin stuff, but I can't help but want to share some preliminary results from a project that I'm doing with WDFW that's on these indirect effects. So I want to spend a little more time kind of unpacking some of these indirect effects that invasive species can have. So this is a species that I didn't know about until last year, a new invader in the lower Columbia, and that's the Siberian prawn. And they are currently spreading up the lower Snake River. We think that they were released from ballast water. And I'm helping WDFW, Andrew Murdoch and Matt Polasek with some analysis on this. And they combine with other introduced species in the lower Columbia, specifically shad. Shad are now the numerically dominant fish in the Columbia. They're anadromous fish, and their juveniles are super abundant in the lower Columbia and lower Snake River. And both these are pretty small. They're smaller than your finger. So and of course, in the Columbia, we're thinking a lot about what could their effects be on native salmonids, which are ESA listed. And those salmonids, of course, swim downstream through the Columbia as smolts that are about the same size of these critters. And so where past literature, if you read a paper on prawns, the potential effects that they really emphasize is competition. So we think these introduced prey species could be having a negative effect on salmonids because they eat the same prey. So maybe they're competing with them for small invertebrates and zooplankton, and that's going to cause, less foraging opportunities for salmon. We're looking at a different effect in this work, and here's some context for it. So these are counts at a dam bypass in the Lower Snake River, and what you can see is that these prawns got to the Lower Snake River in the late 2000 aughts. I'd normally, if I was referencing time, I would make a pop culture joke here, but I'm so out of touch now that I don't even know what trends went out of style in the lower aughts. But so we see prawns come on, we've seen an increase in shad, but following these, we've seen the establishment and an exponential increase in walleye, a voracious top breeder in freshwater ecosystems. So that begs the question, how are these invasive prey species affecting this invasive predator and the effects that it has on ESA listed salmon smolts? So the reason why we might think there would be indirect effects is because salmon and these invasive prey now have this sort of shared predator, and they could potentially be interacting with each other through that shared predator. And you might think, well, an optimistic hypothesis is that now walleye have to decide between eating these invasive prey and the native prey. So maybe they've switched and they only eat this invasive prey, and that dampens their predation on salmon. So let's hope that's the case. But Andrew and Matt collected year-round data on walleye diets in the Lower Snake River, and that allows us to see how they forage on these alternative prey. And in addition to native salmonids, we have information on their diets with native lamprey. And so I'm going to show you a couple, just real quickly unpack a couple graphs. The top is the frequency of occurrence of these different prey items in walleye diets. So that's the percent of walleye that had this prey in their diet throughout the year. And then on the bottom, it's the same idea, but now it's showing the amount of that prey that they had. So not just was it present in the diet, but how much of it. And these are really clear results. So the first thing you see is, it doesn't look to be the story we were hoping for. So this is spring when juvenile salmon are migrating through the system, and salmon are blue here. So salmon are providing the biggest rations of the year for walleye. They are chowing down on salmon in the spring. But then what we see is that juvenile shad have an asynchronous phenology, and they're available in late summer and through the fall. So they're providing the second completely asynchronous pulse of foraging for walleye. And it's harder to see in this plot on the bottom, but you can see up top, the green, the dots at the top of the graph are Siberian prawn. So they are the most common prey item in walleye. Even though they only got there in the late aughts, they are now the most common prey item for walleye. And they eat them year round, but particularly in between these pulses of shad and salmon. So what this suggests, unfortunately, is that prawn and shad are providing sort of the perfect complementary foraging opportunities to salmon because they occur asynchronously to salmon throughout the year. And what appears is probably happening is that they're having these negative indirect effects on salmon because they provide these complementary foraging opportunities that are propping up the walleye population. So this is just further emphasizing that if we stop just at trying to research and understand direct effects, we may be missing some of potentially even stronger effects that these invasive species are having through indirect interactions. Okay, so now that I've probably allocated too much time to introduction because I wanted to share that example, let's get to the main course and the Klamath Basin. And here, we're again going to be talking about indirect effects, but also thinking about water temperature and disease. And water temperature and disease are kind of they pair together well because oftentimes the effects of a pathogen on a host are temperature dependent. So the Upper Klamath Basin is it's a really exciting place to work for a variety of reasons. And you might think, oh, it's one of the reasons because we just had one of the world's largest restoration projects go down with the removal of main stem dams. But there's a variety of other reasons. For example, it has just this tremendous diversity of habitat conditions that I think is captured in these two photos. Everything from these turquoise springs that you can see flying over in a plane. It looks like a little pocket of the Bahamas when you fly over. These turquoise waters to waters that are neon green. For example, at Upper Klamath Lake, there's massive algae blooms that actually support commercial harvest of algae. And across sort of any habitat dimension you can think of, gradient, valley confinement, temperature, elevation, slope. We see just this dramatic range of habitat conditions. And our research group, one of the things that we focus on a lot is trying to understand how redband trout integrate across these habitat conditions through their different life histories or through their behavior throughout the year. And the reason why we've been studying redband trout so much is partly just because we think they're a really cool fish, but also because they're important culturally, economically, and to conservation. And that's because they provide the last remaining subsistence fishery for the Klamath tribes. They support a commercially and like locally important recreational fishery. And as the only abundant salmonid upstream of the dams, and one with sort of similar ecology and physiology to that of southern latitude, steelhead, and chinook, these fish provide a valuable proxy for anticipating how future runs of salmon and steelhead could use the basin and some of the constraints and opportunities that they're going to face. So anything we learn about redband trout could apply to salmon and steelhead in the future. So what we've learned in our past research on redband trout in collaboration with ODFW, the Klamath tribes, and others, and with additional funding from the CASC, is that they grow large. The fluvial and fluvial life histories tend to grow large in seasonally warm habitat like the lake that are really productive. But they use other habitats at different times of the year, and particularly they tend to spawn and rear in colder tributaries. But while they're the only abundant native salmonid remaining in the basin, they co-occur with two invasive salmonids. And those include brown trout, like this one that's part of this fishery in the Wood River, a tributary to the lake that's pictured in the top, and then brook trout. And these species can be quite abundant in the tributaries to Upper Klamath Lake. And you might think, well, that shouldn't pose any threat to redband trout, because I don't know if you can tell, this redband trout here is the size of about a coho salmon. It's about 8 pounds, which is not atypical for the fish in this basin. This is a pretty tough predator. It can take down a 6-inch blue chub in the lake. So why should it care about brook trout and brown trout? But of course, if we take into account the ontogenetic niche and the fact that it's not always a big bad bruiser, but it has to hatch and be a tiny juvenile, then that changes what we think. And Jordan Ortega, as part of his thesis research, was studying the diets of the resident salmonids in these systems. And one of the things he found, I'm going to show you a nasty picture, so look away if you don't like seeing fish guts. But he found that brook trout and brown trout were super abundant in many of the tributaries, and that if you sampled their diets, they were packed full of juvenile redband trout, and they were more abundant than redband trout in these systems. And there's some anecdotal evidence from ODFW surveys that redband trout are in decline in the places where brown trout and brook trout are abundant. So these are potentially, significant predators and competitors for native salmonids and for future runs of salmon and steelhead. So then a key question is that our partners expressed to us is just trying to understand what is their distribution across the basin. And when thinking about, so there's tens of millions of dollars being spent on restoration in the Upper Klamath Basin. So we could help them, our partners, be more strategic if they could understand where is the risk of these invasives the highest, and where are there places that are good for redband trout and future runs of salmonids, but not as good for brook trout. So one of the things we're trying to do is understand what is the what are sort of the temperatures that favor redband and not the other invasive trout. And through local knowledge of district biologists and some publications based on that are largely based on their expertise. Basically, we've gotten the picture, and so that work is by Benjamin et al. At Jason Dunham's lab. But we've gotten sort of the impression that these invasive salmonids are restricted to the tributaries, and surprisingly, they seem to have a similar thermal niche, where both brown and brook trout, don't do well once temperatures get above 18 degrees or so. And the reason why I say that is surprising is because while we know that the salvelinus, the char, like brook trout and bull trout, while we know that they are cold water specialists, we don't usually think of brown trout as being cold water specialists. For example, my in-laws live in Montana. We go and visit them in the summer. And if you float down a river there and maybe do a multi-day river trip and you fish for trout, The trout that go the farthest downstream into the warmest habitats are brown trout. They're the ones that are downstream mixing it up with smallmouth bass. For example, hoot owl angling restrictions go into effect in Montana when it gets to about 23 degrees C. So that's much warmer than the temperatures that seem to be associated with a decline in brown trout in our system. So basically what I'm getting at here is that the sort of anecdotal expert opinion suggested these fish weren't in the warmer water. So what we want to do is first corroborate that and test whether that is the case with a more robust data set, but also try to understand why is that the case. And talking to local experts in the system, as well as folks at OSU who work in the Klamath Basin and study disease, we developed the hypothesis that, and this is kind of a mouthful and I'll unpack it, but that pathogenic parasites are constraining the distribution of these fish and that they're keeping them out of these warmer habitats where that would potentially be, we would normally think of them being within their thermal niche, but that they are kind of shrinking the thermal niche of these fish. So a pathogenic parasite is just a parasite that has a negative effect on the host. And there are a whole lot of them in the Klamath Basin. One of the folks at ODFW that we work with once jokingly said to me, anytime I catch a trout in the Klamath Basin, it kind of looks like it's dying. And what he meant by that is that there just are a lot of naturally co-occurring parasites in the basin. And sometimes you'll catch a redband trout, and it looks like Swiss cheese from all the lamprey marks. You have parasites like Larnia, which is actually an introduced parasite, black spot, and the most well-known parasite, the one that's most likely to make news in the basin, is C. shasta, which can generate massive fish kills. And C. shasta, there's a lot of complexity to C. shasta that I won't unpack in this talk, but it is it is native to the basin, and the salmonids in the basin co-evolved. They co-evolve with C. shasta. So while it can kill them, it's not automatically going to kill them. But there is some gray literature and local expertise that suggests that it is lethal for salmonids that did not co-evolve with it, and that it does kill out of basin fish stocks. So this was the basis for the hypothesis of our research, which is that C. shasta or potentially other pathogens that they are more virulent for these non-native salmonids, and they restrict their distribution across the landscape. So we think that and it's funny. When we first started the project, I made this slide, and I think it shows you how fast Game of Thrones has kind of left the pop culture consciousness. Because now I forget what this was even about. I forget who these characters were. But our hypothesis is that that disease has a positive indirect effect to these native salmonids because it has a negative effect on invasive trout. So it's basically the enemy. The idea is the enemy of my enemy is my friend. And I'll have to rewatch Game of Thrones to understand why this slide once made sense. All right. So what were our methods for testing this kind of complicated hypothesis? And I really think that a study like this would have been really hard to do without like a $10 million budget, even just a decade ago. And that's because it is really difficult to quantify the landscape distribution of any taxa. And it's especially difficult for things like disease. But with new tools such as eDNA and spatial statistics, we're increasingly able to characterize the landscape distribution of different taxa, oftentimes with the same sampling protocol. So what we did in this study is we collected water samples, triplicate water samples at 64 sites across the Upper Klamath Basin. And the fieldwork and tools were developed by the co-PIs on the project, Jordan Ortega and Julie Alexander. And then we used spatial statistics to take our observed data and then make predictions of the distributions of different fish and disease taxa across the basin by developing these SSM models that had covariates, including the one we were most interested in, which was August stream temperatures, but also additional spatial data layers that you can get, like accumulated base flow index, stream slope, and watersheds is just a random effect. And so what we did is we tested whether the presence of pathogens alters basically the relationship between temperature and native fish species. Okay, so I'm gonna, and there was, we are kind of just getting and analyzing, we just kind of finished our first kind of round of analyses and we're wrapping up the project. So these results are still gonna be a little bit preliminary. And there's a lot to unpack here, but I'm just going to kind of give you the kind of the high level information. And so here, what you can see is these are sample sites, and if they have dots, that's where fish were detected as present or diseased. And then the colored lines represent the extrapolations from our spatial stream network model with all of the covariates and the results that we saw. And so what you can see here is redband trout, and a key point I forgot to make is that the DNA meta barcoding that we did that allows you to kind of get the whole community with one water sample. It has the taxonomic resolution of genus, and so we're going to be talking about genera, but that worked out fine for this project because redband trout are the dominant Oncorhynchus, and because of the dramatic decline of bull trout, brook trout really are the dominant Salvelinus. But redband trout were widespread. And salmotrota and so brown trout and brook trout were confined to the headwaters. So this formalized with more robust data the hypothesis from expert opinion that these species were really confined to the headwaters. And they're not really in the lower portions of rivers like the Sprague. And we saw really so we focused on C. Shasta, but Julie Alexander kindly ran additional parasites for us. So we looked at C. Shasta and another myxozoan parasite. We looked at nanophytes, and we looked at ick. And so if you look at the spatial distribution of these different pathogens, each one was really different. So the myxozoan parasites, including C. Shasta, were really only found in the lower portion of the basin and around Upper Klamath Lake, whereas nanophytes was upstream of the lake and then in the Sprague River, and then ick was around the lake, but then more in the Sycan. So they each had a different spatial distribution. And one thing I want to emphasize here is that as we were designing this project with our partners, one of the things that they emphasized is that each of these maps in itself was something that was helpful for them. So they weren't too worried about, they weren't only concerned with what was the broader hypothesis about indirect effects, but just having information on the spatial distribution of invasive species and pathogens would help inform them just about the spatial patterns of risk to native salmonids in the basin. Okay, so it's hard just looking at these maps to understand what the relationships were between these distributions and the distributions of salmonids. So what I'm going to show you next is what the model results showed. Okay, and so here the different rows on this graph are for different salmonid species. So we have redband trout, brown trout, and brook trout. And the columns are for different covariates. And I'm going to focus on water temperature, this left column here. So what this shows is the relationship between stream temperature in August and then the probability that fish were present during our August sampling. So this is pertaining to summer. And at the top, you can see redband trout. There was a weak sort of effect of temperature where they're pretty much present in all sites, but they start to have a lower probability as you get to the warmer sites. But there was a really strong drop off for both brown trout and brook trout, at relatively cool temperatures. And then we want to ask is, well, does that relationship change depending on the presence of these pathogens? So each of these lines is for the presence of a pathogen. And what you can see here is there doesn't actually seem to be an effect of the myxozoan parasites in green. They're mapping up with the no pathogens line. But for brown trout and brook trout, you do see that this relationship drops off faster in the presence of nanophytes. So it suggests that pathogen is potentially constraining the thermal niche of these invasive taxa and could be having a positive indirect effect for the redband trout. So now I'm going to show you an odds ratio plot, which looks at the effect size of each of these variables and its statistical significance. So if it's on the left side, it's having a negative effect, and on the right side, it's positive, and the further over it is, the stronger the effect. And if the error bars cross 1, that suggests it's only marginally significant. So what you can see here is at the top of the plot that temperature was really significant and had a strong effect on the invasive species. They're only in the colder habitats. And then if we get down here to the bottom of the graph where we have the pathogens, what you can see here is that you're really only seeing a high effect size, a strong negative effect for nanophytes in brown trout and brook trout. But there's there's a lot of variation in the effect size. So even though it's a strong negative effect size, it's sort of marginally significant. So as I said, we're just getting these results in and starting to write up our work. And our next steps, in addition to the remaining analysis we have to do, where we're looking forward to trying to move forward this project, is additional sampling to increase our statistical power for example, for making that analysis of nanophytes more robust. Using more targeted DNA methods so we can get information for individual species or estimate relative abundance. With that additional data, we could do joint species distribution modeling. And then lastly, we're excited about moving forward to complement this observational species distribution approach with experimental or observational work on quantifying host-parasite interactions and how they change across temperature gradients. All right, so that concludes my talk. I just want to thank the Northwest RISC, funding from the Northwest CASC, our collaborators, and thanks Eva and Darcy for arranging this talk. And I'd be happy to take any questions if I have time.
Thanks, Jonny. And we are running up into Ian's time, so we're going to go ahead and I will introduce Ian now. So happy to welcome on Ian Tattam. Ian, you can go ahead and start screen sharing while I just give a quick introduction. Ian's Eastern Section Science Program Manager for Oregon Department of Fish and Wildlife. And he's an Oregon native who didn't catch his first steelhead until age of 10, not for lack of trying before age of 10, though. He went on to receive a BS in Environmental Science from Portland State and an MS in Fisheries Science from Oregon State, and wrote his theses on Oregon steelhead for both degrees. Ian has worked on Eastern Oregon steelhead and salmon research for the past 23 years, 7 with Oregon State University and 16 with ODFW. So Ian, I'll turn it over to you.
Awesome. Thank you. Thank you, Eva, for having us. Can you see my screen okay?
Yes, thank you.
All right, let me see if I can find the laser pointer tool here. All right, looks like I got it there. All right, thanks everyone for joining. So yeah, I'm going to try to mix a little bit of country music in the sense of beer and gasoline don't mix with some steelhead monitoring data we've been collecting in the John Day River Basin for the past 10 years. I've been a small component of this project, really. Lindsay Sapella, Liz Blackburn, Eric Rook, and Herb Winters have spearheaded the data collection, analysis, and implementation of this, but I'm here to present today. So I want to first give everyone a little bit of overview to summer steelhead distribution throughout the Pacific Northwest. These green lines here are the summer steelhead distribution layer throughout the Pacific Northwest, both inclusive of both endemic and non-endemic, right? So there's some, non-endemic distribution shown here in the Upper Willamette and other areas of the western parts of Oregon and Washington. But the majority of this is the endemic distribution of steelhead, summer steelhead, premature migrating steelhead throughout Oregon, Washington, and Idaho. And then the John Day River Basin is outlined in gray here. And so just trying to use this slide to make the case that the John Day represents a pretty substantial chunk of the pie of total summer steelhead distribution throughout this tri-state area. So John Day watershed is outlined in gray there. Each of those green dots are sample sites we've used for other projects. For this presentation, I'm going to zoom in on the 2 primary areas, which I'll show in a second. But I want to go through 3 different things here. One is a little bit of life cycle context and background. Secondly, some observational data. And finally, data from a field experiment we did at one of these sites. So moving on to a little bit of life cycle context here for John Day steelhead. Again, here is where they are. They have to migrate downstream and upstream over 3 dams on the main stem Columbia River to get back to the John Day watershed. For a period of time from the mid-aughts, and I need Jonny to give me a cultural reference here, but from 2004 to 2012, we conducted basin-wide surveys for natural origin spawner abundance throughout the John Day River. Those estimates range from about 5,000, low of 5,000 to a high of about 15,000. Concurrent with that time period, wild summer steelhead counts at John Day Dam, which is this point right here, immediately downstream of the John Day River mouth, range from about 55,000 to 163,000. So again, thinking about our slice of the overall pie analogy, about 9% of the entire Columbia summer steelhead run, give or take, was entering and spawning in the John Day Basin in those years. So a little bit of brief background about the John Day River and the smallmouth bass impacts that I'm going to be talking about here. First, smallmouth bass. They, of course, were introduced in the Columbia River system in the early 1900s. But prior to their introduction into the John Day River by the Oregon State Game Commission in 1971, this falls right here, Tumwater Falls, we're standing here at about River Mile 10. So 10 miles upstream from the Columbia is this waterfall and narrow spot that was believed to be a barrier to upstream migration of bass. A couple things to note there is that one, the John Day was inundated up to about this point right here in 1968 by the construction of John Day Dam. So now the hydrology of this site is probably different now than it was prior to 1968. We have some evidence that bass are migrating upstream from the Deschutes River into the John Day near this point where the photo was taken, and also a couple snippets of bass tagged in the Columbia, their pit tags being detected upstream of Tumwater Falls at pit arrays about 10 kilometers upstream of this point. So that's sort of a long way of saying that even without this direct introduction, we may, in this day and age, still have been in a place where bass were able to successfully expand from the Columbia into the John Day. But of course, they're there now, in part due to that introduction in 1871. What we're really concerned about is bass migrations into steelhead spawning tributaries for several reasons that we'll get into here. And, to step back a little bit, there have been a number of studies in the main stems of the John Day, primarily the North Fork and the Middle Fork, led by University of Washington folks that looked at the interaction, competition, and predation of bass on steelhead and those dynamics. These studies generally found that there was not a direct predation effect nor a huge competition effect between bass and steelhead in the main stems of the North Fork and Middle Fork of the John Day. In part, we kind of hypothesize now that was because there is a longitudinal thermal sorting effect in the main stems of the rivers, right? Where the native salmonids are tending to be upstream in the colder areas, and then you have this nice kind of progressive gradient in temperature where it rises, and as it's rising, it's less desirable for salmonids to be there, but more desirable for the warm water smallmouth bass to be there. So they're moving into the warmer areas where salmonids are naturally dropping off, and you only have a little bit of overlap here where they're spatially in conflict if you have this kind of long thermally changing longitudinal gradient. But in contrast to that, we focused a lot of this work that I'm going to be talking about today on tributary streams, where some of which don't have that longitudinal gradient as denoted by this figure here, where we have sort of a discontinuous longitudinal tributary stream network and then a really small area of cold water, which is juxtaposed to the warm main stem. And so we thought that this might create different outcomes in the steelhead bass competition or interaction effects that we needed to investigate. And so today I'm going to talk about primarily two sites that we've investigated. One is 30 Mile Creek, this red circle in the lower John Day Basin. The second is a series of tributaries in the lower North Fork, a series of tributaries to the lower North Fork of the John Day, sort of right in the center of the John Day River Basin. And over the past decade or so, we've been pursuing two tracks of tributary steelhead research here. The first is an observational track. We've done this both in the North Fork John Day tributaries, where we've looked at presence and overlap, and also in 30 Mile Creek, where we've evaluated stomach sampling for evidence of predation of steelhead, fry, and parr by bass. And secondarily, we've done an experimental track where we did a before-after control impact study to remove bass from a section of 30 Mile Creek and evaluate changes in steelhead abundance post-removal. So I'm going to jump into first the observational comparison here. And this was a project in the several tributaries to the North Fork of the John Day River. So located in this northwestern corner of Grant County, Oregon here. The North Fork John Day that University of Washington monitored previously is flowing from your upper right here. It flows around and comes down and then exits downstream to the main stem over here. We investigated 3 tributaries as part of this study. One was Big Wall Creek here, flowing from the left down into the North Fork. The second was Cottonwood Creek, flowing from the lower right down into the North Fork at the town of Monument here. And the third was Rudio Creek here on the left. So in this situation or this study, we went and monitored an abundance of both bass and steelhead, primarily their presence, but also their density, estimated density at each of these sites. So each yellow dot is a sampling site. The yellow dots that are ringed by this red shade is where we actually physically saw or captured smallmouth bass concurrent with steelhead. So you can see their bass have moved up from the North Fork a little ways into Big Wall Creek, further into Cottonwood Creek, and not at all, at least not detectably, into Rudio Creek based on our sampling methods. And so I think the key takeaway from this study is this comparison of juvenile steelhead density in sites that have smallmouth bass absent, which are noted by these open circles, and smallmouth bass present, noted by these solid black circles. So first of all, for the smallmouth bass absent sites, sort of similar to what Jonny showed earlier, we see this declining trend where you have higher density of juvenile steelhead or omicus in cooler water areas. And then as you get, this slide moves a little further over to the right than Jonny's slide, right? I think his slide stopped around 20 for the temperature effect, but this one goes all the way out to 30, which is basically 30 is about the upper incipient lethal for an instantaneous sample of steelhead. This is the 7-day average max. And similar, the open circles are pretty consistent with other densities of steelhead throughout the John Day Basin. You get about a steelhead per meter at these ideal temperatures, and then it slowly declines to 0 when you get to about 28 degrees for your 7-day average max. In contrast, we did not see that sloped relationship for sites with smallmouth bass present, right? We're at, your medical professionals would call this asystole here. We're at this flat line where, we really have very low density of steelhead at these higher temperatures, but worryingly, we also have low density of steelhead at these 20-degree temperatures, which at sites without bass can support 0.5 or maybe higher steelhead per meter. So this difference right here is potential direct impact, whether it's from predation, competition, or the synergy of both of those things on steelhead. Okay, so concurrent with that monitoring of density, we also collected some observational evidence of direct predation on steelhead by bass, primarily in 30 Mile Creek. And I won't dig into this too much, but I guess a couple of key takeaways here is that this predation can be highly episodic. So, it's kind of like fishing, right? You got to be there at the right place at the right time. And if you're a day late, you're more than $1 short in terms of seeing that bass are eating steelhead. But even given that episodic nature, it can still have a potentially a really big impact on total steelhead production. So shifting over to the experimental side of our of our work here. We're going to jump into 30 Mile Creek and talk about our bass exclusion and removal there. So a little bit of context here. This is 30 Mile Creek flowing from the right down to your left here where it joins the main stem of the John Day. There's 3 primary perennial sections of 30 Mile Creek noted in by the blue lines here. The black dotted lines are ephemeral sections that support surface water from about January through June in typical years. And so because there's no flow close to the surface here during the summer, or not enough flow, these sections really have no surface, no riparian vegetation. So when stream water is flowing on the surface through these areas, that stream water is receiving almost 100% solar access during the spring months. And so we'll get into that in a second, but that's an important part of this story. But we use these perennial reaches here as a control impact study, where this mouth perennial reach was our impact site because bass are able to get into this area. Bass generally don't access these upper areas, so we use that as control sites for steelhead abundance. And we did a multi-year study where we had 3 years of allowing bass to do whatever they wanted to do. And in 2020, we installed a weir to exclude bass right here. And we basically made this a steelhead only zone here in impact reach, where in prior years, bass had access in and out of that reach. So circling back around to the temperature profile here that I mentioned a second ago, the blue line is the lower John Day River temperature, so the main stem temperature. The pink purple line is the temperature of 30 Mile Creek in the reach where we excluded bass. And so starting at Julian Day 68 here in roughly late March, right? Or I guess that's early March. Water temperature trends in these 2 streams are tracking each other fairly closely until you get into April here. And again, in April, water is flowing on the surface through the ephemeral reaches. And as air temperatures start to heat up, sun angle rises, you have more daylight, higher angle of incidence. The water temperature in lower 30 Mile Creek heats up dramatically because of those ephemeral reaches. And now you have, during April, you have potentially a 5 or 6 C difference in water temperature, with 30 Mile Creek really rising dramatically. And it stays much warmer than the main river until this connection between the ephemeral and perennial sites happen. When the water goes subsurface into the perennial, then the temperature there drops dramatically. But during the spring period, it's artificially elevated over a natural stream temperature profile. And this is occurring at a time right in April when the main stem John Day is gradually beginning to cross 10 degrees C, which is an activity threshold for bass. So those bass are waking up from their winter hibernation under a rock or a bush in the lower John Day, tootling around looking for slightly warmer water to where they can operate more efficiently. And hey, great, they find this much warmer water in 30 Mile Creek, zip into there, which unfortunately that their immigration kind of co-occurs with the prime period of steelhead fry emergence, which is this vertical band here. And so then we have this direct conflict where they're together in space and time, right when steelhead fry are emerging from reds and are at max vulnerability to predation. So for our exclusion experiment in 2020, we operated this weir. We were able to create a 76% reduction in bass abundance in the impact site. And then these are the results for steelhead par density. So in the control reaches, again, these are the, this is the control phase in the pre-monitoring period, the impact monitoring period, and the post-monitoring period. We really see no difference, about 2.5 steelhead par per meter throughout the experiment. Whereas in the treatment reach during the pre-phase, there's about 1 steelhead par per meter. In the impact phase, when bass are removed and excluded, that jumps up to about 8 par per meter. And then in the post-monitoring phase, when bass are again allowed back in, drops down back down to about 1 steelhead par per meter. So clearly there's a dramatic immediate boost. And so this is where we excluded bass in the springtime. And then those young of the year that were not consumed by bass during that critical spring window survived to the par phase that fall. So they survived the first 5 or 6 months post-emergence at a much higher rate without bass presence. But of course, that leads us to the question of where do we fall on the scale of compensatory versus additive mortality. If bass predation of steelhead fry was compensatory, and those fry that we save by excluding bass and grow to the par stage are just later consumed by birds or other fish, or die of natural mortality due to lack of food, then removing that source of mortality, of course, wouldn't have a total impact throughout a longer period of the life stage. There's been some evidence for both sides of this in the literature for steelhead predation. So to try to stretch this out as far as we could, we looked at steelhead smolt survival to John Day Dam, again, the first dam they encounter on the Columbia, and tried to estimate the impact of our experiment on steelhead smolt production at that site in the Columbia. And we've got pretty similar results here as well. So for the control site, you're consistently producing about 500 to 600 steelhead smolts down to the Columbia in both the pre, the impact, and the post phases. Whereas the treatment reach, where we excluded bass in the pre-phase, we're at about 500 steelhead smolts. In the treatment where we impacted, the impact phase where we removed bass, we increased production to about 3,500 smolts. So about a 3,000 smolt increase. And then we're back into about the 500 smolt production realm in the post phase. So this is some evidence that exclusion of bass carries through at least to the smolt phase, and kind of suggests that, again, at least through the smolt phase, predation by bass leans towards the additive mortality side of the spectrum for steelhead in this example. So in conclusion, circling back to our apple pie analogy here about how impactful is production of 3,000 more smolts from 30 Mile Creek. For context here, the annual average smolt production from the South Fork John Day River, the entire South Fork John Day River, is about 25,000 smolts. So this one experimental manipulation produced about 12% of that abundance in 30 Mile Creek. Umatilla River Basin production is about 50,000 smolts annually. This experiment produced about 6% of that in basically a 1 kilometer reach of 30 Mile Creek. So I think this is some evidence that this predation impact is having some consumption of our overall pie of steelhead in the interior Columbia. For management applicability, just at a really like a back of the envelope level here, we spent about $5,000 in personnel and material costs to exclude bass, really in a kind of a experimental fashion, granted, from 30 Mile Creek. The costs we spent on the research, monitoring, and evaluation of this far exceeded the actual work spent constructing and installing a weir and removing bass in this experiment. For context, hatchery summer steelhead production is about $5 per smolt when they arrive at the first dam, to be apples to apples with our data, our estimate of smolts at John Day Dam here. So it would have cost us about $15,000 to replicate this bump in production within a hatchery. So in this case, potentially there's some cost savings by doing work and increasing natural production versus going into a hatchery. The unknown here is that the scalability of bass exclusion really remains a question. How many places is this occurring across the landscape where we could have a benefit by doing some sort of exclusion or removal during the spring period? And of course, that's a hard thing to know because, as I mentioned about the episodic nature of predation, you need to be at a lot of sites for a lot of time in the key time window in spring to really see if that direct predation and competition is occurring. And as a final thought, obviously habitat restoration and rehabilitation is the key to providing space for steelhead to be separated from bass. But while we're going through the phases to get from, intermittent stream that warms rapidly in the spring now to a perennial stream that stays cool, we may need to consider some supportive bass control, sort of a la places where we do supportive breeding in terms of a hatchery supplementation program to help populations get through this current phases until we can restore habitat and restore migration conditions. Some of these places in John Day may be better suited to supportive bass control than supportive breeding strategies. So that's what I have for today, and just want to thank everyone for joining. And I'll end with this is mural art in downtown Condon, Oregon. So my son's giving it the thumbs up, and we're hoping that this work in tandem can help provide and keep steelhead in the streams, not just on the walls for the long run. So thanks all for coming today, and hopefully we have some time for questions.
Awesome. Thank you so much, Ian. And we do have a few minutes for questions, so I encourage you to put any questions you might have for our speakers today in the chat box. If we don't get to all of them in the remaining time, we'll try and get those answered via text that we can include in our recording description. And we do have a question from Anthony in the chat saying, are any of the tribes involved with predatory fish removals in these smaller tributaries? And is there a desire to eradicate smallmouth bass entirely?
Yeah, good question. I guess part A, the tribes have been supporting our work in 30 Mile, but they have not been directly involved in the removal of predaceous fish there. I think that's something they would probably have interest in long run. Part B of the question, I think the horse is kind of out of the barn on that one, and I don't think it's practical, feasible, or really socially palatable to completely remove smallmouth bass. But there is a need, I think, to control and manage their distribution across the landscape, so we can have, steelhead in one spot and bass in another. Bass in areas where they're not co-occurring with critical life stages of steelhead or salmon.
We do have a comment from Olivia of love the extension of conclusions to natural production versus hatchery, not to mention consideration of multiple other factors to give us recommendations for complex real-world scenarios. Both talks were fascinating. Thank you. I wonder maybe if, Jonny, you could talk a little bit about just some of the where you and Ian have crossed paths and where your work intersects a little bit.
Well, we kind of co-advise. We've co-advised a couple of students, so we get to interact a lot. And I'll just kind of jump in and riff here. Like, I'm really fascinated with the temperature dependence of predator-prey interactions. Like as it gets warmer, like does one becomes favored over the other, and getting to work with Ian's group on 30 Mile, it was fascinating but kind of depressing, because I always thought that salmon smolts were safe from bass until it got pretty warm. And I'll never forget, we were puking this smallmouth bass, and it must have been only 12 degrees C that day, I think. And nothing would come out. And I was like, wait, I'm a pro at fish and puke. Let me help, or puke and fish, let me help out here. And I was like squirting water in its mouth, and it coughed up this big smolt. And anyway, so I don't know if that answered the question, but it's been fascinating for me to see that the kind of the some of these relationships that we think about between temperature and invasive species are more complicated than we think, and just they're hard to study like experimentally, but they deserve more study.
Yeah, I think what Johnny's getting at is there's a lot of heterogeneity in temperature in 30 Mile, and it can go up and down. Like you're looking at maybe a 10°C diel variation in the springtime, right? And so you can, if you're there one day, it's 10 degrees, you're like, well, bass aren't going to impact steelhead, but the afternoon before it was 20 degrees and they're just cruising around doing what they want. And that, yeah, it takes a huge amount of effort to be there enough to see that.
Well, I want to thank you both for being here and thank you everyone. We do have an exit poll that RC is launching, so if you want to take a moment to answer that, and otherwise add any final questions to our speakers in the chat, and we'll get them post facto to you all. Thank you again, Ian and Jonny. It's really
Thanks,
cool.
everyone.
Thank you.